Hello again. So today we're going to derive several simple rescue models and then try to go to a bit more complicated genetic basis for the rescue. Next lecture, I will try to talk on how we make a link between the environmental change itself and the rescue process, because so far I've just said there is a rescue mutation. It has this value. Here is what happens to it. So we're doing really the population genetics of the rescue and demography of the rescue. But next lecture we'll do more about some ecology in it, if you want. So yesterday we talked about. I told you I would present two possible scenarios of rescue. The first scenario will be from a mutation selection balance state initially. We saw a bit yesterday what it would look like, but I'd like to get back to it. There is a classic way of presenting mutations arising and invading populations, which is called the Muller plot. The Muller plot usually is like this. You have some dimension in the vertical axis, and the horizontal axis is time. And these are genotypes. And usually you say that this is dominated by the wild type genotype. And at some point in time, somewhere, one of the wild type line produces a mutant that then can invade. And you have things like this, and then you have some other mutant that may appear and disappear. This is what mutants look like in Muller plots. So this is very useful to conceptualize a bit what happens, especially with asexuals, where you have mutants hindering each other's fixation, things like that. But when we are doing rescue, we have a number. The number of genotypes here is not stable because we have a number of individuals. Sorry, here is not stable. So these are really well. Sorry, I should write it. You see, my eraser is now not erasing the whole page. It's wonderful. So these are individuals really, because here at the beginning they are all the same genotype. So in a rescue process, we would have something like this, but the difference is that at some point the environment will change. So we'll have the permissive environment before and the stressful environment after that. It's a discrete, abrupt change in the environment. The other difference is that we're going to have to represent the fact that the population size is changing. So we will do what, I don't know, it has no name. I've just realized I could explain you things for this class. So I don't know if it's been named somewhere before. But I would call it a demographic Muller plot. And in this plot, we also plot the number of individuals, but the number of individuals changes over time. So we start with a given large population, and for some time, we are stable like this. And actually, if we are dealing with the model with mutation selection balance, we are stable for a very long time in the past. So we've been like this all the way to infinite time in the past. And at some point here, the environment changes. And there is a stress. This means that the fitness of the genotypes who are here, all of them, because this is now very polymorphic, this fitness changes suddenly, and in the case of the wild type becomes negative. So I will represent just because I'm saying that most of the population is composed of the wild type at mutation selection balance, assuming that the mutation rate overall is quite small. And the wild type, there is two scenarios. The first one is going to be to say that we have a bottleneck, because as we said, if we are in a large population, we must do a bottleneck. I'm going to erase pieces of it. Sorry, my cat wants to get in and is not going to stop until I open. So I will open because he's the real boss in this house. So we do a bottleneck. No, this is not going to be possible. If it's viewing, I'm sorry. We do a bottleneck. No. Okay. This is the thing I must work with. Okay, so you see the conditions right now. My apologies. We're doing a bottleneck and then the population size decreases. And if it was only the wild type, it would go this way. So we are looking at these, we're going to say they are log sizes. So I have a straight line going down, and this slope is my wild type growth rate, which is negative. So that's what should happen if nothing else genetic was happening. And now we're going to consider for the moment just that only what happens in the past and has consequences in the stress environment. So only the standing variance process. In the mutation selection balance, there are a bunch of genotypes which are at low frequency. So they are really kept at a steady appearance disappearance because it's a finite population, a very large finite population. But for the problem, we can consider that they are at some low stable frequency. And there are several such lines because they rise independently. We could consider there are several such lines like this. And due to the bottleneck, only one such line, here I just draw 3, only one will be able to contribute to the rescue because this one was not sampled. This one was not sampled. And this one has two things that happens. First, it has a bunch of the copies that are in this mutant class. They get lost by stochastic fate, demographic fate. The reason is that before, in the past environment, we had no death happening or we neglected it. And so each genotype arising was kept with probability 1. It's just that its frequency was going smaller and smaller due to selection. If it was at mutation selection balance, so it was deleterious in the previous environment. And now this genotype changes environment where the wild type now decays, so it does not hinder the growth of the mutant. And there is a death process which makes it decay. This death process means that every copy that is in here, each of those copies, they stand a given chance to, in the first generations after the environmental change, to die out stochastically because they underwent more death events than birth events, or at least the net demographic outcome, stochastic demographic outcome of this given cell, this given individual was negative. So one of the lineages is lost, another is lost, and one of them, one of them avoids this extinction, and now it can increase. It's actually straight lines because it's log scale demography. And this guy is now invading, and that is a standing variance evolutionary rescue. So we must focus on this process first, and then we will consider other possibilities, which are lines that will occur in this phase during the decay process. So first, we need to know the amount of lines that are present at the onset of stress. As we saw, if in the initial population, that's scary. If in the initial population, the frequency was at equilibrium, it was equal to mutation rate over cost. So that was the frequency here. of my mutants. And I'm drawing N0 individuals. This is the size here. I'm drawing N0 individuals, and I'm sampling them, each of them with probability P infinity. So this is a binomial. The number of lines I get is a binomial. That would be the probability of choosing, well, these sets of mutants among all the possible mutants that were here in the initial base population. But I now have to weight this probability for the future fate of the lines, because out of all those lines here, only a part of it, a proportion of them, will make it to avoid stochastic loss. So avoiding stochastic loss is called establishment. And there is a probability of establishment, which I will call PE for establishment. This is the probability of establishment. And for the moment, we will not detail what it is. It depends only on the lines on the lineage, not on whatever happens around it, because we're in a density independent stochastic process. So it depends only on the lineage. Typically, it depends on its growth rate and the variance, the stochastic variance of its demographic process. We will see some more detailed model of that later. But for the moment, we just say there is some probability of establishment for this mutant. And so I have to weight the success, the number of success here, the number of mutants, by the proportion of those that will, in top of that, avoid stochastic loss when rare, so that they can contribute in the long term. This probability of establishment, it's the probability to be a non-zero population starting from a single copy. But it depends on time. Initially, of course, your probability is 1. You're sure to be here after a few seconds after you were here before. But if you look for long enough time, you reach a stationary value for this probability, an asymptote. And this asymptote happens to go quite fast. So usually for the purpose of modeling, even at a demographic scale, we may not do it, but we can model the problem as the establishment probability over infinite time. The reason is that anyway, what counts is that in the infinite time, what rescue will be is that in infinite time, you will be there. So we just need the infinite time probability. So that's why there is just some probability of establishment, which depends just on the lineage properties, but not on time. And so the success probability is just, we now have the number of, so so I need some room, so I'm going to go down. We had that the resistant mutant count follows a binomial N0 P infinite, where P infinite was our mutation selection balance equilibrium. Now, the number of ER, which are evolutionary rescues, is a binomial, but we are weighting by the probability of establishment on top of that. So we are just waiting by this further fate that will happen. And then we can take a simple limit, because we assume that this is small and this is large. So we take the Poisson limit that we saw yesterday, and we have that this is roughly, this is roughly a Poisson. A Poisson distribution with value N0, P infinite, PE. And so that's how we get our number of future rescues. So now, what does it mean to have a rescue from just this process without considering whatever happens later on during the decay? Well, it means that it's 1 minus the probability to have no rescue. Because as we said, we're a density independent process. So whenever you don't get extinct, you explode. So we just have to compute the probability that no such mutant is there. So this is the probability of extinction. So the probability of rescue then is just the probability that probability of rescue is 1 minus the probability that MER equals 0. And this is from the Poisson distribution property, 1 minus E minus lambda SV, which is the rate that is here. The rate being equal to the expectation. So this way we have a theory, a quite simple theory for the probability of rescue if there was just standing variance. Now, of course, during the process, some other mutants may arise during decay. So I'm going to represent again the same thing. My past MSB.
There's a question. Nikhil, do you want to ask?
Sorry.
Yes. Oh, yeah. Can I ask now?
Yeah. Yeah, of course.
So in this, are you just considering one mutant for now?
You mean one type of mutant?
Yes.
Yeah. So basically, another advantage of the Poisson limit that we can derive in this case, because mutation rate is small, because of this small p infinite and large and zero. This Poisson limit will be handy, because if you consider that there were several different types of mutants, which is of course the one thing you want to do because it's the reality of the biology, you would just have to implement the. So I can spoil the result. You will have a Poisson process on space, the space of growth rates, the space of types, and you will have a mutation rate to this particular type with a given growth rate. It has a given cost. So this cost is connected potentially. A given mutation with a given growth rate in the future, in the stress, will have a given cost in the past. Okay. These two may be related. And it will have a given PE of R. Okay. So you will have some on any spatial growth rate, you will have that. And you know that in each class, this is Poisson.
Yeah.
And so by the property of the spatial Poisson process, your total number of rescue will be just the integral over all the possible types.
Yep.
And that's because we have a Poisson.
Yeah, I follow the argument. But now the next immediate question which comes is that after we do the sampling for one time, it seems that the number of types remains n naught. The total number of, not the type, the total number of individuals remains n naught. But I thought that the population size would change.
We do the sampling once at the point of the bottleneck, which is really what is done in some experiments where you have a large population, then you do a stress and you sample from this population to plate it on Petri dish or put it in a tube. And so the sampling is really a discrete event that happens once at the onset of the change of the environment. Actually, empirically, if you work with bacteria, you have to do it because you are, at least it's more comfortable to do it because you have to remove the former medium to put the new medium.
So
it's just once. And then the population size later on, it becomes decreasing, but we're not sampling from it anymore because for the moment we're dealing only with the standing variant guys. So after we have done the sampling, all our exercises stopped because we have just modeled the future of those lines by the establishment probability, but then it's done. If those lines who are sampled manage to in the future avoid stochastic loss, they will do a rescue.
I understand. Thanks.
Now we're going to do what happens to the new mutants that appeared here in the new environment. So that was the first part, that's the standing variance. And then we're looking at the process. So again, it's the same draw, blah, blah, blah. This is my change in environment. And now I have another type of mutation which can arise during the decay from the wild type. And these mutants, many of them also get lost. So they rise and die by, again, the same process of stochastic loss. But some of them will arise and invade. And this is now an ER DN for de novo. This is the classic term. The one before is standing variance because it's already there at the onset of stress, and the other appears de novo after the onset of stress. But you have to keep in mind that all that is mutation, appearing de novo at some point in time. What is important here now is that the mutation process is a bit different from the one before. Before, you have a regular rate of mutation. You have a arisal of mutation all the time at constant rate from the wild type. You will note that in everything I'm doing here, we're ignoring the mutation rate back from the wild type, from the mutant to the wild type. We're ignoring multiple mutations, which we will deal later on, and we're ignoring epistasis. We're ignoring a lot of things. We don't need to. Well, we're not ignoring epistasis because we're dealing with the one-step problem. So there's no question about epistasis. So this problem here is really the same as before, because you can consider a stunning variance just as you would consider a mutant. So if I put them green, a stunning variant is just a de novo mutant that arises at time 0. The problem is exactly the same. So you have the same parameters because these are the, ooh, this is exotic. You have the same parameters. It's the same mutant. You have a PE here. And this PE applies to the mutant the same way. Because it was the PE for the stunning variance model was applying to the mutants in the stress environment. So it's really in the same environment. You also have, that's a question, an open question. You may have the same mutation rate to the variant. This is a more open question because if you change the environment, you may change the mutation rate. So for the moment, I'm gonna allow for this to be different. I'm gonna say that there is a mutation process that is different in the new environment, because in the past environment, we were having somehow a stable population. If you were considering bacteria, for example, they would be in stationary phase. They would have almost no division happening. So you cannot consider that mutations happen at division, but still there is mutation. And it's more difficult to study empirically, but there is mutation. And mutation comes probably, at least that's what I've heard, from errors happening when you read the genome. And you read and you kind of make the book different by just reading it, because you deploy the DNA and then you can insert mutations sometimes. Here it's a bit different. Most of what's happening that can create a mutation is during a division. So then we will have a process of mutation that is going to be birth rate of the wild type times a mutation rate per division. So this is a probability. This is between 0 and 1, and it's a probability of mutation per division. And then, of course, this is for every division. So we have to multiply by the number of divisions that happen at a given time, which is BU N of T. And this thing changes because N of T is here. It's going down. So you have time N of T, well, log N, huh? And you have a lot more mutations produced now than you have in the end. That's why most rescue mutations arise early on in this time. That makes things difficult to estimate if you want to know if the rescue came from the past or from the future or from the de novo, because actually the ones from the past, they are like if they rose. Actually, it's rigorously like if they rose at time 0 exactly, but they look like they rose in the past because there may be multiple copies of those. So if there are many copies, they look like they appear later on in the past. So they seem to appear around here. And from the point of view of the regrowth curves, they seem to have a reason somewhere here. And the de novo, they seem to have a reason somewhere here. But the problem is that most of them arise not here, but around here, because there is a lot more mutations happening in this time. So their origin of time is very narrow. They look like they appear at a time very comparable to the stunning variants. And on top of that, because there is stochasticity in the initial growth, it blurs the signal from the origin, the true origin of those mutations. So it's quite difficult to detect if they are standing variant or de novo just from a curve like this, from where they seem to appear. Anyway, so now this is ugly, so I'm gonna switch. So now we need to go with the same model, but now we have mutation occurring as a Poisson process for the reason we saw yesterday. So we know that by time t, by some time t like this, the number, the mutation, well, sorry, I'm gonna say it otherwise. The mutation process to resistance is a Poisson process, but it's time inhomogeneous. But that's not a problem, as we saw with the Poisson, because it handles time inhomogeneity with no problem. And the rate, the instantaneous rate, is B, birth rate of the wild type, times U, times the current N at time t. And that's your mutation process to resistance. Now, the same process to ER is a conditional Poisson process. It's conditioned on establishing. And we know that because of that, this is a Poisson process too, with rates. So that was lambda R. We have now lambda ER equals this, the rate of the native Poisson process multiplied by the success probability, by the conditioning probability. So again, you see all we saw about the Poisson process is really, really used all the time in these models. And that remains a Poisson process. Yeah.
Yeah. So this assumption of inhomogeneity of Poisson process, is it related to this density independent assumption that this lineage?
Not really, because it's due to, I think, if you have a density dependent decay, like initially you decay faster because you are hindered by the high density, and then you decay density independently, you still have a Poisson process with some rate that is changing over time. The key is really to assume mutations occur as a Poisson process. And that's what makes it then become a inhomogeneous Poisson process, which remains a Poisson process. So it's really the assumption of a Poisson process that makes things that makes the computation the way we do it. If we have, for example, I did a model, I can talk about density dependence in the end quickly, but I didn't do too many simulations on it, etc. So, but you can do a process of decay of the wild type that is not exponential, but density dependent, logistic, and or something else, and you can compute your rate of production of mutants in this new demography, because it's all, the equations here are not saying anything about the fact that N of t is driven by some density dependent process. It's just N of t. So if you are able to compute the integral of N of t over time, you're fine, whatever the thing is. So because we have this, the number of ER dn, because of the Poisson process property, we know that by time t, by some time t, big T, is going to be Poisson distributed with parameter the integral of the Poisson process over time up to the time we're looking at. So from 0 to t, b, u, n of t, p. So we know, again, that we're going to have a Poisson number of ER. And now we can, of course, make this integral explicit, because we're saying that n of t has a simple exponential demography, which is, I think yesterday I said, I prefer to do it this way, actually. So this is a positive decay rate of the wild type. So the growth rate is minus this. So you have n of t equals rd. And then if you compute that, you will see that the integral has a given value. There is a trick to compute it quite simply as a memory. It's not very rigorous, but as a memory trick, it's useful. You say that dn equals minus rd n dt, and that means that if you put this on the other side, you have n equals minus dn dt over r. And that means that your integral, so you have b, u, pe, they are in factor, because pe again doesn't depend on time. It's a property of the genotype, and it's an infinite time limit, so it's a constant with respect to time. And you want to compute this integral. In fact, you can write it, so n dt. This thing, you can write it as minus 1 over rd from 0 to t of dn. And so that means that you're looking at dn integrated from a time to another. So this is really minus 1 over rd, the value of n taken at time 0 and at time t. And so as there's a minus, you can do something like this, rd n taken at time t and at time 0. Why do that? Because it's very simple then. The integral of n from 0 to t is just equal to 1 over the decay rate, now the decay rate itself, times the initial value you had minus the value you have by time t. So with this, you know the proportion of mutation of lineages that is produced up to any time. Again, remember, this is a number of mutants produced. It doesn't tell you anything about the size of this mutant class. And that's important because the number of mutants in the population will not be Poisson. Because even if you have one lineage arising, then if it goes up, it's going to be large. And in the end, of course, it's a very large number, which is not consistent at all with this theory. This theory is presenting you the number of mutant lineage. So MR here, this is a number of mutation events, if you want. It doesn't tell you how many guys there is in each lineage. It tells you how many lineage there is. And we just need that. The reason is that we're going to compute the extinction probability. And extinction happens if no lineage is there. So we don't need to know what happens to those lineages if they are here, because if they are here, rescue happens. So we have computed our rescue probability. So we just compute extinction probabilities really all the time. So we are looking for the 0 class of a Poisson number of lineages. And if no lineages arose that is destined to be established, then there's no rescue. There's no rescue. It's going to be extinct. So that's why we don't have to deal with the demography of the mutant lineages. So this is fairly simple. Actually, from that you can derive the density of the times of appearance of the mutants that produce the rescue. So rescue mutations happen to arise proportionally to with exponential decay. They arise as an exponential.
I'm a little bit confused here. Sorry. You said that we don't have to worry about the establishment probability of the mutants that arise that are going to rescue?
We do. We don't have to worry about the sizes they have, their actual size, but we have to worry about their establishment. Do they get extinct or do they make it through to be present at infinite time?
Right, but then how do we, because you said that we just subtract the probability of, well, basically does a mutant arise at all? And then 1 minus that is basically a probability of rescue. Mm-hmm. But then the probability that it arises is not the same as, is not accounting for the establishment probability, right?
Yeah, it does, because it's here we were dealing with the process of resistant mutants. So this was a Poisson process with rate, birth rate times mutation probability per division times the current size. And then we added the conditional probability of establishment here.
Ah, okay, got it. Yeah.
So
thanks.
by adding that, we corrected for, we kept all those resistance that are destined to not get lost.
Right.
So we did correct for establishment.
Thanks.
So in the end, we have
question. Yes. So for this time t, the answers will depend upon this time t, right? The capital T that you're writing.
This
So
one, yeah.
shouldn't I think of the fixation time? I mean, it's destined to fix, but the time scale that matters is the one in which fixation happens. If this capital T is much smaller than fixation time, then again the rescuing probability or number of lineages to survive will be essentially zero, no?
In fact, what we're doing, why I'm giving you this time is just to see that you have access to a bit more than just rescue probability. But in fact, here is my decay of the wild type. It's just that I can look at this particular time, and the production of mutations of ER mutations will be equal to the difference in size between N0 and N of T multiplied by BUPE. But of course, the main application of that is to look at this time, which is really the time where N of T, because there's no time where an exponential is zero, but that's the time where N of T is negligible compared to N0. So in this, we're going to really use the MDN is Poisson BUPE times 1 over RD N0, because we just say that this is negligible compared to this. So whenever we are long enough in the extinction process, when we are through the extinction of the wild type, that's really when we want to know if rescue has happened overall. It's just to say that if you want, you have access to the number of rescues that arise in this area of time, in this one, in this one. But the key computation is this one.
The computation is fine. I'm just wondering, I mean, the question is
that would be lambda dn, sorry.
Yeah, what is this capital T? I mean, how do...
This is just one time. So we were just saying that...
Right now it's general. I mean, it's...
Okay, you want to have the expression for the time. Yeah, okay. So the time, if you mean that what is the time where the extinction is complete here, I think it's log n0 over r or something, I think. No, it's not log n0 over r. It's, I never know it by heart. Sorry. It's probably log n0 over... I don't know, because I always use the end result, which is the one where time is large enough that we have finished the extinction. So,
no, so if you're going to say that T is basically log n naught by R or log of n naught R by R, then I understand what is this T. Because it's basically the time to fixation. Is that the time scale that you are referring to, or is it something else?
So the time scale I'm looking at is the time scale where the extinction has fully happened. So I guess for the fixation probabilities, that would be the time to fixation, although it's a bit different. I have to do my... but this time, to compute it, typically, if you want to compute it, we don't need to compute it, right? For the end result, we just need to say the time is long enough that this thing is now negligible compared to this one. And then we have our rescue over the whole extinction. Okay, so we don't need to compute it to get the rescue. We just need to be large enough that this thing, we are around here, that extinction has de facto happened. Because of course, the log population goes on and on and on. And there's never a value where it's zero, because it's an exponential approximation to extinction. But if you have the way to compute this time is to say, typically what people do is you say, okay, I have N0 E minus RDT. This would be my population size. That's the graph I was showing. And you say, what is the time where it is equal to 1? So that 1 is negligible compared to R0. And if you do that, which is the way they do for the fixation probability also, they compute the time until you reach 1 minus, your P is you reach 1 minus Q, something like that. I don't remember. But you don't compute the time to 0. You compute the time to one copy, one copy left. Typically.
So
that's the same. You compute the time to just one cell left.
Yeah. So it's basically that argument, the same usual fixation time type argument. Okay, fine. Then it's clear.
But you don't really need to make a choice about this, because here in this case, which is different from the fixation probability time, you have something that is big here and that is driving the result. So you don't need to be very precise about this as long as it's long enough to be small compared to this, which is really essentially saying that most of the extinction process has happened. Okay. But if you want the time, you compute that, so that would give you. What is it? It's. I'm very bad at computing.
No, I think it's all right. I think I
but
understand.
you see the idea. You solve that.
Yeah, sure.
You take the log base that gives you minus RDT equals
minus log and not.
Yeah, it's minus log and not. Yeah, oh, here it is.
Yeah, no, it's okay.
So that, and then, yeah.
So it's all right.
Yeah, so that would be this. Okay, so just to summarize the two things we had. We have, oops, not the right color, sorry. We have standing variance rescue. There are MSV, Poisson, U over cost times PE times N0. And this thing is the parameter of our Poisson number of rescue from standing variance. We have Denovo rescue, which is also a Poisson. And this rate, so after all the way to extinction, the rate is B times U times PE times N0 over R decay. And that will be lambda dn, a rate of the Poisson. So in this case, what do we interpret? Of course, you can see that this is proportional to the mutation rate to the resistant mutation you're looking at, divided by its cost in the past. It's proportional to the establishment probability in the future of this mutant, and of course, to the number of guys you put in the stress initially. And in the de novo, it's proportional to the mutation rate overall per unit time, which is really the birth rate of the wild type in the stress times the mutation rate. It's proportional to the same establishment probability, the number of guys you put, and to the decay rate of your population. So what happened is that if you have very costly mutations, even if they are quite frequent, you won't see them from the standing variance. And you will see much more of them here, because you can assume that if U is large here, then little u should be large here. If you are in stress, that makes B equals 0. For example, an antibiotic that just destroys any capacity to divide, then there is no mutation de novo, not at all. This is the basis, the implicit basis actually, of the Luria-Delbruck analysis. They are just ignoring whatever mutation happens de novo, which I think is wrong. Well, I know is wrong from some experiments where you can measure it. You can see the biomass increases after the stress, but it's sometimes a very good assumption, because some stresses make your birth rate be 0. So that's where there was a point in looking at the mutation process in the stress environment as a per division process. The establishment probability is going to be the same, and 0 is going to be the same. So it's all going to be scaled with this. And now this, of course, tells you that if you're in a stronger stress in the sense that you decay faster, you have less de novo rescue. Of course, because you have less time, less overall, less biomass producing the less cumulative size of the wild type to produce those rescue mutants. The other thing we have from that, so of course, we have the total probability of rescue, because the probability of rescue overall from the two process. The advantage is that it's going to be the probability, it's going to be 1 minus the probability that MSV plus MER equals 0, because you have no lineage able to rescue in the standing variance and no lineage able to rescue de novo. And then we use again the properties of the Poisson, which is that if the two things are Poisson, then the sum of the two, sorry, I did wrong here, MDN. So as those two are Poisson, the sum of the two is Poisson. So this thing is Poisson distributed with a rate equal to lambda, which is lambda SV plus lambda DN, the sum of the two rates. So it means that we can compute our probability of rescue. It's going to be 1 minus E minus lambda, where lambda is given by lambda SV plus lambda Dn, which is really, we factor by N0 by establishment probability, and we have U over C on the one hand, plus BU over RD on the other side. If you want to do a simple birth death process, the decay rate here can be written as the death rate minus the birth rate. So you can write it all in terms of the death rate and the birth rate of your wild type in the environment, in the stressful environment, this thing. The other interesting thing with that you can come up with is that it's a property that when you look at two processes, or you do two conditions, two Poisson counts from two things. But you count, say you put a blue dice and a red dice. Well, the dice are not Poissons, but you can know the proportion of each type in the end result. And here, that would be the percentage of ER in your ER that is from standing variance or de novo. And that thing is just the ratio of the rates. So here I need to say it. In the notebook, I said the from de novo versus SV. So that would be P, probability that your rescue arises from a de novo is lambda de novo over the sum of the rate. It's the contribution of the parameter of your Poisson you're looking at over the sum of the Poisson. So that gives you simple expressions because this thing cancels out, and you end up with a simple ratio, which I put on the notebook, but I don't have here. So it's going to be B, sorry. It's going to be BU over D minus B on the top, divided by U over C plus BU over D minus B here. And then things further simplify if you assume that your rate of mutation per unit time, U, in the past was the same as the rate of mutation per unit time in the future, BU. But I don't make it here because I think it's usually not really true. So for cells dividing versus being in stationary phase, it's not very true. If you were considering rescue in a discrete time model, you could make this assumption, I guess.
Could you explain this stationary phase idea a bit? I missed something. Why do you assume they're in stationary phase before the stress occurs?
Because typically when we do a culture of bacteria and we study rescue, we study, say, we want to stress with antibiotics. What we do is we have our number of cells here. We grow them from a single colony. They go up, and then they reach some stable. The sugar is lacking because we grow them in a closed system. So the sugar is missing. By the end, they have consumed all the sugar, and they enter what is called a stationary phase here. And this thing is a subject of itself in microbiology. What happens there? There's a set of things happening around here when they switch to stationary phase. It's really a phenotypic switch. But anyway, in this phase, they are at stable size. And that's when we will typically do a dilution and then put the antibiotics. And then it goes up or it gets extinct. So this stationary phase is a time when there is no accumulation of biomass. And when I measure dead cells too in it, I don't see much accumulation of dead cells much. So if the stationary phase was in fact a set of birth events compensated by death events that compensate each other exactly, so something like B minus D equals 0, but B is not 0, then I would see biomass accumulating and I would see dead cells accumulating actually at the same rate. While I see the number of live cells stable, but I would see biomass accumulating like this and dead cells accumulating over time. And it's not what I see. So the assumption, more realistic, I think, and I think it's known in microbiology, is that B is just 0. So if I say B is 0, I have a problem with my model of mutation happening by division, because there's no division happening. There's no way mutation can happen.
But all of this assumes that you are in stationary phase when you're doing this experiment. You don't have to be, right? I mean, to have
no, you don't have to be, but if you're
right.
considering a stable population,
right,
you have somehow to be there for some reason. And
no, but in
the
a game
one
of
way
catch,
we are
for
there
example,
with bacteria, at least the one way we're there is this. Now, in the natural environment, you could consider that there is something that puts out the individuals regularly, like in a chemostat, and that kills them from external reason, and that they are reborn. So then you would have B minus D, and B is positive, D is positive. So some external factor kills them and compensates exactly their birth events. But that's tricky because you need something that exactly stabilizes. But of course you can say that. And if you say that, then you have a mutation rate BU, the same mutation rate BU in this phase as in this phase, provided that from this environment to this one, B remains the same. And it's just that D rises to some higher value here.
Actually,
but
I think
B can also go down because of the stress.
Well, I mean, working with bacteria, I feel like that is actually not a terrible assumption. No, most of the times, if we're growing cultures in a nice log phase growth, we don't necessarily have to wait for that long to get to exponential phase.
Oh yeah,
and the mutation rates don't have to change terribly either. I mean, yes, with antibiotics or
okay. You're considering this kind of scenario, which is the one I'm going to do now. So I think you're considering you start with this and you grow them. Say you even grow them staying in log phase. So there's a lag and then you grow them. And there you put the stress and you kill them.
Yeah, something like that.
You're considering that. But that's a different model because this model, there's no way this thing is in mutation selection balance. This thing is now a Loya-Delbrück experiment type. So we're going to do this one now. It's different because when we were saying, before we were saying the key thing for the strong invariance, we were saying that the frequency of the mutant was U over the cost. And that was based on a stable mutation selection balance, which requires times of the order of U over C, I think. I don't remember. Long times. I won't say something false. So I don't know what time it takes, but it takes long to reach mutation selection balance. And so you don't reach it and everything in the Loya-Delbrück, for example, is not a mutation selection balance theory. This kind of theory is the one you can find in Orr papers on rescue and Orr and Unckless. 2014. I will give some references at the end of the lectures, but this is a fairly famous one. And the Holt and Gomulkiewicz also, which are in sexuals, they also assume this kind of initial situation.
Right.
But the first rescue models, these are from the 80s. These are from the years 2000s. But the first rescue models, they come from Luria and Delbrück, which was a very important experiment in microbiology, and that you know of, I guess, because you do bacteria. And this was in the 40s, I think, the initial paper. And it's actually this experiment, the fluctuation test, is actually it's a rescue experiment, and it's actually maybe the only evolution results that has received the Nobel Prize for medicine, because they got the Nobel Prize for this. But by that time, they were not doing it to study rescue. They were doing it to know if mutation was something happening, at least that's what I've read. Mutation was something happening independently of its effects. So the way we model mutation now is to say that mutations happen and then whatever they produce is selected later on. But originally, when people discovered mutation, and especially mutation to resistance to antibiotics or to phages, there was an option also to say mutation is just a plastic response to the fact that you're stressed, and it's being like switched in when the stress happens. And they proved by the fluctuation test, testing the shape of the fluctuation, that the model considering that they arise randomly and then they are selected was the best one. So I'm going to switch to this experiment. So now the new experiment is a different situation in the past. Instead of having a mutation selection balance, which we cannot have unless we have a stable population for a long time, we now consider that we are growing from a single cell or a small population all the way like this. But that's a different experiment, and the conditions, the ecological conditions are very different because this is not at mutation selection balance. And now, so did I answer your question, Deepa? Is it clarifying?
Yes. Yeah.
Do
Thanks.
you not agree? Yeah. Now this new situation is different because then stress will happen. So this is what would happen with the wild type. So that's in permissive condition, and that's in stress. So here we may or we may not, depending on whether we consider that the change in the environment has affected the mutation rate itself. But then we have here some BU rate of mutation per divisions here. And we have some rate B prime. Well, I used to call this B, so I'm going to call it B. And I'm going to call this one B star U. And we have BU here. The rate of mutation, that's a rate of mutation per unit time. And B is U is probability of mutation per division. And this is the same U. We could also say U star. It doesn't require this to be equal. Now, what happens here is a bit different. We have mutations for the standing variance process. Of course, for the de novo process is exactly the same. We have not changed the assumptions about the stress environment. The standing variance process is different because now mutations arise in a growing population and they have a given time to invade, and they actually are less hindered in their invasion than they were in the stable population, where in fact they cannot, there is density dependence in this stable population, and it's impeding. The density dependence is what makes you have minus CP1 minus P. This thing happens even if you don't have, even if you don't have density dependent demography, you have this kind of thing term in the change in frequency of a mutation. But because just frequencies cannot go beyond one. But if you have density dependence, the thing is that at some point you cull the population to create your initial population. And this culling makes you have a low probability to be sampled if you are not frequent. So it's kind of hindering the mutant to be in large numbers. Here it's different because there are some mutants that arise early on. And if they arise early on, they will be numerous, even if they don't grow so much faster than the other one. And if they arise later on, they will be less numerous. And most of them arise late, and some of them arise early. Just by the same mechanism that we had here. There are less cells here to produce mutations than there are later on. So the whole principle of the Luria-Delbrück experiment is to study the statistics of the number of guys you get here by the end of this part. They don't consider any de novo mutation. They consider that whatever they have here, when they put it on a petri dish with antibiotics, for example, the number of colonies they get are the number of resistance they had here. In fact, it's not true. First, because but that's been I mean, the fluctuation test analysis is an ongoing subject in modeling and statistical analysis of experiments. So there's a lot about it. The very nice and informative data is the sizes of those mutant groups. Many of them are very small, and some of them, like this one, are large, and they are called jackpots. It's like cases where your petri dish is filled with colonies. The thing is that for our problem, we don't need to know all this. All we need to know, again, is the probability that one of those, that none of those lineages will be present and able to do a rescue later on, because we're just considering the probability of rescue. We're not considering the distribution of the sizes that are here. You have to know, though, that at least that's my point, that when you take there are two limits in this analysis. When you take a bunch of cells, so you sample from that first, huh? So that's modeled. You sample from it, so you really are taking not the whole. Well, some experiments you take the whole culture. Some of them you can sample, so you can also do that. So you will have lost some of the mutant lines, but you will still have some of those here. But when you put them on the Petri dish, first there is a sampling, but then there is still the problem of being established. When you grow on a Petri dish, it means you've grown from, if I draw the Petri dish here, this is agar. You grow from a single cell that spreads and produces what is called a colony. And that's exactly what it means to be established. That means being a big population starting from a single individual. So the lines that are here, they are not resistance. They are evolutionary rescues. They are established. But what we are dealing, so it means that we have a way in these experiments to estimate the established numbers. Now, the problem here is that we don't have a stable process of mutation production, just the same way we didn't have in the decay phase. So now what is done is there are several models. The distribution of those types depends now on the stochastic growth of each mutant. And this stochastic growth can be many things. You don't necessarily, they may have non-exponential distributions of waiting time to division, they may, etc., etc. The assumption we're going to make for us is only that the process here is a pure birth process, so that whatever the time you take to do a division, there is almost no death. That means that every lineage that appears remains present. But we could also wait by an establishment probability in the preculture. Because we will have, again, a Poisson number, we will make this Poisson assumption because it's going to be a Poisson process here. We're going to say that the number of standing variants is going to be Poisson something with some rate. And because of that, we know that the expected number is going to be this rate, because that's a property of the Poisson. So by properties of mutation being rare, we're going to assume a Poisson number of lineages able to do a rescue. By the Poisson property, we're going to say that the expected number is going to be lambda SV. And now, if you do that, you can, I have to skip this computation actually, because otherwise I won't be able to talk about the rest. You can just take the dynamics of the mutant. So that, let's say N of T is the wild type count, M of T is the mutant. You have the change in N is B times 1 minus U, because you must not mutate, times N, plus, minus B U N of T. No, I don't need that, sorry. It's just this. It's all the guys that are born from wild type and that are not mutants. And the mutant count is BUN. This is the new guys produced by mutation from N, and plus BM, the birth rate of the mutants, M, because that's the growth of those mutants. So if you do that, you have a system of ordinary differential equations, and it's deterministic. So it's really a deterministic approximation. It's a rough treatment of that. Okay, and from that, you can compute the total. So you can solve for this, and you can compute the total amount of guys you have at some endpoint. And this endpoint is T is such that N has grown from an initial to a final N0. So basically, this time is just this. You start from an initial and you finish with N0. And if I did the math right and if I read it right, because there are some errors even in papers, so that I used in the past. So if I did this right, where is it? It's in the notebook that I've sent yesterday. I'm going to open. Sorry, I should have had it in front of me. Why is it not here? Voilà. You work out the, so in the notebook, I write the derivation. You can work out a number. So first, the time it takes, so the time t takes to grow from this single cell to single cell or initial cell size is, as we had it before, it's log the final size over the initial size divided by the birth rate of the mutant. You can show that it's the time you take for the mutant, for the wild type to grow all the way, whereas in fact the true size is the wild type plus the mutants. So this is wrong. We stop the culture when the wild type when the whole population reaches some level. But if your mutation rate is small, in fact, if you write really n plus m, the sum of the mutant and wild type, and you look for the time to reach some given value, you see that the things are closed. And now you look for the number of guys you get at that time. And this number is really the expectation of MSV, because we are using deterministic ODEs. And these ODEs are linear with the states. So it means that they are giving you the exact expected outcome. When it's linear, the ODE gives you the exact expectation of the counts. So M of t and N of t are really giving you the expected numbers and not the actual numbers, but they are without bias. And so this thing will be our lambda, or rate of the Poisson. And this is now depending on all the parameters. So the final size and 0, the initial size and initial, the birth rate of the wild type, the mutation rate, and the birth rate of your mutant. But if you say that U is small and that B mutant equals B times 1 plus epsilon, and you say that this epsilon is small, so you say that really the mutants have a growth rate that is not too different in this environment from the growth rate of the wild type, then you have a leading order for that. And it gives you this: MSV equals U times the population size times a log factor of growth, the log of the factor by which you grow the culture, times 1 plus epsilon over 2 times log and 0 over 9. So here you can see that the difference, what would be the cost of the resistance here, in our previous interpretation, the cost of resistance would be minus epsilon B wild type. So there is a cost. Whenever epsilon is not 0, there is a cost, and it could even be positive or negative. There's no need for it to be negative or positive. You have it in your expression. But in practice, you can neglect that effect of differences in growth rate between your wild type and your mutant in the growth period, whenever that thing is small compared to 1. And if you plot the expression here, you will see that, so it's in the notebook, but you will see that if you plot the threshold, basically, even when your factor of growth is of the order of 30, so it's less than 30. So it means you're growing by 30 logs. So it's a huge growth. Even then, you would need an epsilon of more than, you could afford an epsilon of more than 0.5, which means that you could afford to have a birth rate of your mutant that is 50% less than the wild type, which never happens by random mutations in a permissive environment. I mean, it's not seen. Apart from a lethal, you get things that are of other order. So that means that, and you never grow by a factor 30 in log. I mean, that's 10 to the 30. So, or e to the 30 factor growth. So in practice, we can in fact quite ignore the differences in cost. And in the end, that's MSV roughly is U N0 log N0 over Ni. And so it's interesting because it means that we don't have to account for differences in the birth rate in the pre-culture phase. So we don't have to know it to be able to measure, to predict the cost, the distribution of these numbers. It's different from what happened in the previous, where we had to account for the cost, and we had a ratio U over C. Here we have just U for the mutant. And now we have to correct for the guys being evolutionary rescues. So this is going to be sorry. These guys are going to be Poisson with this rate. So let's call it lambda SV R for resistance. And the MSV ER are just the same, but after correcting for establishment in the future. So you multiply again by probability of establishment. And so in the end, you have your rescue probability that is given by U N0 log N0 over Ni, which is really by how much your population has increased before the stress, times probability of establishment. And that's lambda SV. We have the same. Sorry.
Can I ask a question?
Yeah, of course.
What exactly is F in the, when you said F is less than 30?
I'm sorry, I didn't hear it well.
What is F in the, when you said F is less than 30, what exactly is F?
My connection is bad. What exactly is
F
was the factor of growth. By what factor you're growing? Sorry. It was. Be careful that I didn't do a mistake in my computation. Let's check it here. Yeah, I'm doing an error in what I'm saying, so I must apologize. It's, yeah, if F is the factor, but what I'm saying here is wrong. You made me realize because I computed that yesterday. F would be the factor by which you're growing this. And maybe if I can share, the screen on the notebook. F is the factor by which you grow your culture. And so to be able to neglect the effect of differences in growth rate between your wild type and your mutant in the initial preculture, you need that thing to be small. So you need basically that epsilon, so the difference, the relative fitness difference. Epsilon is just B wild mutant over B minus 1. So you need the difference in birth rates between the two types to be small compared to 2 over log F. So, and what I was saying is not right, because I was considering it was just F and not log F. So it's plotted in my notebook, but well, I can maybe show you that, but then we won't have time to do the multiple mutants. Kavita, is it better that I switch to the multiple mutants, or I detail a bit this log F, this standing variance issue?
Yeah, I think if it's a smallish point, maybe people can look it up and then discuss with you tomorrow, or have a separate session. Maybe I think you wanted to cover more things, but I have one question, which is, I think I still have not appreciated the difference between these two cases that you said, in which there's a mutation selection balance in the first phase versus when the expanding population. So it's really about the amount of genetic variation, standing genetic variation is different, right? Is
Yeah.
that sort of point? So which one is better for the population, the expanding population or the mutation selection balance?
It depends on the cost in the mutation selection balance versus I don't know. I've not checked it because the cost is also there, because the cost is here. It's in
So it will
here.
depend upon this, right? Where I stop the expansion and what's the
Yeah. So
rate
I would
and
have
the
to
cost.
do an analysis of it, because the cost is there. The mutation rate, you need to make an assumption on how the mutation rate in a stable population is or not the same to the mutation rate in the growing population, because you note here that the birth rate of the wild type has disappeared. So this is a mutation probability per division, whereas before we had a mutation rate per unit time. So it's a bit different. And then there is this term. So
Previously you had what? U by C?
U was a big U, and it was a per unit time.
Yes, it was U by C into N naught. Was that?
Yeah, U by C and not. Yeah.
Okay. So now you have.
Now I have that, and if you want to connect it to C, C would be if you consider a pure burst process, it would be BM minus BM. B minus B star. The white. I called it B star. Sorry. The wild type birth rate minus the mutant birth rate, that would be C. And here epsilon is, so then epsilon is C over B star. That's epsilon. So you see, it enters really differently, and this is this you would have to compare it in the small cost linearized term, but that's probably not crazy. And you would have to see, depending on the parameters, what gives you the largest lambda in the end. I've never done it.
Yeah, but
it's just that the idea is that we can compute an explicit outcome, and there is an effect of the cost in these two, and it should happen because, and that's what makes it more logical. It should happen because if you imagine that your mutation selection balance has happened because you have let your population grow indefinitely for infinite time in a growing context, then this log F here, this becomes infinite.
Yes.
And the costs start to play a role, and it should, because in the end you're with an infinitely growing mutation selection balance population. Because of course the theory for frequencies does not depend on the population growing or decaying. It's the same. It's just a deterministic approximation, but it does not change if your population grows or decays. That's the point of looking at frequencies, that it's independent of the actual sizes of the population. So your population, if you let it grow to infinity, it should be a mutation selection balance population, and we should get back to the other results. But then you need to account for this infinite growth and then the bottleneck, because you cannot be at N0 then, because you're letting it grow to infinity and then you're sampling from that.
So I think
maybe I can look at the computation and tell you tomorrow.
Yeah, can I just I think maybe I misunderstood some point here. Can you just go a little up where you were drawing the Muller plot for this case? Yeah. So I thought I will wind up, when I take the sample at the bottleneck, I'll basically wind up taking those lineages which arose early on, because they will get a disproportionately larger weight, and therefore genetic variation would be lower in an expanding population.
Yeah, but there are also many more. There are also much more of the lineages that arose later on. There are exponentially more of them. So it's a balance.
Yes,
the guys from the far past, there are numerous per lineage, but the guys from the recent past, there are many of them. And you have recurrent mutation towards the same type. So there's a distribution of it.
Is it genetic variation is high-ish? I mean, I don't know, compared to what case like that.
Yeah, so this is a known thing, that a population that is expanding tends to have more diversity, and a population that is decaying tends to have less diversity than a population that is stable. So here we see this kind of effect, is that if you are doing the expanding population, you can see that even though your mutations may be costly because they have an epsilon that is not zero, for some time, at least before you become large, you can neglect this effect. So you're really doing as if you were a bit neutral, because a lot of you don't lose so many types when you're expanding. So you're keeping more diversity than when you're decaying. It's the opposite effect, and when you're stable, well, it's the reference, let's say.
Okay, so you think that the standing genetic variation is more compared to the mutation selection balance case in this case?
I think so, but I need to really write down those explicit connection, because I never tried to do it, and see what happens. So I can put it in the notebook and talk about it quickly.
Yeah, that's okay. Yeah, thank you. Yeah.
I also feel like in that case, when you calculate for the mutation selection balance, it would depend on your assumptions about how that population is maintained under that condition, right?
Yeah. And the mutation rate associated with it.
Right. And I feel like thinking about it as a bacterial stationary phase population is a very specific, narrow example in my mind. I would imagine a chemostat is a much better way to think about it, where births are balanced by deaths, and that is what maintains, the steady state.
Yeah, you can. So that's what we were drawing in the end when I answered your question here. This one. So you would have B and D canceling out. So D is really the outflow of the chemostat here, and B is canceling out exactly. That's what the chemostat does.
So when you make the comparison, is what I'm saying. I think that's the better way to
I don't know which is which. It depends on what you're actually doing in your experiment. If you have a chemostat, or if you have a
no, but for a more generalized way of thinking about this, right? Not
the chemostat is a very special ecology too. I mean, where in nature does it happen that you put exactly the amount of food you need to be on the brink of famine, but not exactly there? I mean, it's a really special. And there are many ways of doing chemostats. Like you can put a lot of food, or some of them they are almost starving, but not exactly. Some of them, they put a very strong flow and they are in constant exponential phase. And that's different. And you can think that the mutation rate will change if you're in starving condition or in true exponential phase condition that is canceled out by strong outflow. I would tend to think the simplest is this version, like you have a very strong food coming in and food coming out a lot. And there is just a constant exponential phase status that is outweighed by a very strong outflow of cells, and that allows you to be in exponential phase. And then physiologically, you're in the same context as the later on situation. So that would make, I guess, more easy. But I wouldn't say what is the most realistic, because some people use chemostat to be at the brink of starvation. Some use it differently to be in really an exponentially growing population that is always compensating for the strong dilutions it's undergoing in a continuous time. Some also, so another model we did, there's a paper we did about several scenarios like this. Another model we did was recurrent gross dilution like this. So you can work out the standing variance in this, but it's different from either the stable and the exponentially growing, because you have recurrent bottlenecks now. But you can also work out the math of it. There are several different scenarios anyway. But whatever you do, it will still be true that you're changing the environment from here to here. And whether this changes the birth rate is quite important, because one of the ways in which you start to make the population decay is that you both kill it and also hinder its divisions. So some cases you don't. So I use penicillins where I think I have empirical, there is literature says, plus I have empirical confirmation that I don't change the birth rate roughly. But there are many antibiotics, for example, that change the birth rate. So you will change the net outcome. So I think it's good to keep a different mutation rate from the other, work out the math with these two, and then say, okay, whatever my situation, I can choose a sub-situation where I can set the mutation rate to be the same, the birth rate to be the same. But it's not more difficult to work out the math, allowing for the mutation rate to be different here and here. So I prefer to present the thing with the two, and then people can put those two equal if they want for their experiments.
Sure.
Is it? It's not making the thing more difficult, because you're dealing with this Poisson thing. You're dealing with a different two distributions: the Poisson from the standing variance and the Poisson from the de novo. So it's not changing a lot of your work to do that. I'd like to finish with what happens when you put a distribution, because it's very straightforward, and I'd like to not do that next time. So what happens when I say, because of course, the actual thing that happens, for example, in our experiments, we sample from resistant types that we produced by a standard fluctuation test experiment, and we get a distribution of growth rates in the stress of those types that looks like this. For example, we get something that looks a bit like this. That would be the histogram of the ER mutants we get. So more generally, of course, we need to account for the fact that there is a distribution of the possible growth rates, the possible parameters of the mutants. Because there's not just the growth rate. The growth rate is the deterministic term, but there's also the stochastic variance that counts in the establishment, for example. So what you just do, you use again the fact that it's Poisson. So that's why I troubled you with the Poisson, because it's used everywhere in this whole thing. As it's Poisson, you can go from a Poisson to a spatial Poisson. And now it's on the space of genotypes, space of R, and if you want, other demographic parameter. So now it means that your Denovo rescue, lambda Denovo, it was U, now it's U of R, in the narrow area, in this space. You look at what happens for a given value of R. So it's going to be a mutation rate to R. It has a given, then there will be N0 over RD. And there was a probability of establishment of R. Typically, we will see next time, where we need to make more assumptions about the actual demographic stochasticity, it's going to be, this thing is going to be proportional to R. Like the probability of fixation is proportional to S. So this thing is going to be proportional to R, but we don't go in here. It depends on R. Because of course, a line that grows very fast will not tend to get extinct as much from one copy as a line that grows merely beyond zero. So what is U of R? Well, if you have a distribution of fitness effects, so this F of R, this thing would be a distribution of fitness effects, then this will be, sorry, there was a B in front. There was a birth rate. The birth rate was the birth rate of the wild type, so we don't have to distribute it. It would be B, genomic mutation rate, N0 RD, times the PDF, the distribution of R, times P of R. And that will be my lambda Dn in the class R, the infinitesimal class going from R to R plus dr. That will be this times dr. That's the point of the definition of the PDF. So the overall rate of rescue, because of the Poisson that we've seen on spatial Poisson, is exactly the same, but it's just integrated from 0 to infinity over the distribution of R. I've put it from 0 to infinity because this thing has some value, but it's always 0 when R equals 0. A line that tends to decay deterministically cannot establish stochastically, because imagine it would establish, it would become large, and then deterministically it would die out. So it cannot, and that's a quite general density independent branching process result. It's not depending on the specificities of your demography. And of course, then it has a given value, something here. So I just take it on the distribution of beneficial. So that PDF would be the PDF of only beneficial ones. And that thing would be the establishment probability for only beneficial mutations. And then this thing would be the mutation rate genomic to resistance, because I'm just counting beneficials. So it means that a lot of the theory we have is just this thing, but we just integrate over, so I can put the integral is just here, 0 to infinity. It's just integrated. That's for the N. For the standing variant, well, in the mutation selection balance, we had, if I remember well, N0, U, and then we had this. Again, probability of establishment, but divided by cost. So here, you have a bit more tricky thing to compute, which is the ratio between the establishment probability in the future. That's about something happening in the future. The property of the genotype in the future, and the cost of this thing in the past. So you have to connect two environments, the past one with the future one. So for example, typically people would assume that the cost of a strongly growing mutation in the future is larger. So you cannot be at the same time very strongly resistant in the drug and not costly in the no drug. But there are infinitely many ways to build these integrals depending on the distributions and the model you're choosing for how the two environments connect and how the distribution is produced. That would be for the MSB scenario. And for the other one, the, say, Luria-Delbruck kind of scenario, the de novo was the same, but the lambda SV was U. So again, that would be a mutation rate to resistance. That thing, I call it to resistance, so that I'm just integrating over positively growing over resistant types. And that would be, again, the mutation probability per division to resistance. I had what? N0 log N0 over Ni, which is really the factor by which, log factor by which I grow my culture. And then that would be just F of R. Check if I'm not doing it wrong. Sorry for the. Yeah, you see that if I don't count this, if I can neglect this cost thing, the whole rate depends on nothing but the mutation rate in terms of genotype. It depends on nothing about the genotype except the mutation rate to it. So in this context, you have a simpler expression because this thing, of course, this thing is just one. It's just the whole distribution. And this depends only on the mutation rate to resistance. So that's why you can evaluate by the fluctuation test experiments. One of the methods done to evaluate the mutation rate to resistance is called the P0 method. And in fact, what they're doing is, sorry. I knew I did something wrong. That if I had time, I would say, please, students, answer to me, what am I forgetting? But I'm afraid of what will happen. People telling me, we didn't follow what you were saying since the beginning anyway, so we don't know. I forgot this. We need to count for established mutants. So, and I'll finish with this. The P0 method is just, you do this growth, you take the population, you sample from it, and you plate it on Petri dish with antibiotics. And you look at how many Petri dish have no colony whatsoever. So what's a colony? In the analysis that is done of it, it's considered to be a resistant mutant. It is not, it's an established mutant, but that's not in the classic analysis, it's not considered this way, but this quantity here, I could call it the mean establishment, PE bar, over all resistant possible. And the P0 method is just saying that the probability of a Petri dish with 0 mutant, so an empty Petri dish, is equal to E minus, well, N0 log N0 over Ni UR. Actually, it should have P bar. And so that's a means to estimate the mutation rate to resistance. That's a very classic one. The advantage of this here is that you don't need to use the distribution of the number of colonies you get on other plates. So it means you don't need to make assumptions about how they grow stochastically. We've made none. We just derive the mean, the expected number. And that's sufficient because for the Poisson problem, we just need the expectation. So that's a very robust way to estimate. It's, on the other hand, not a very nice, elegant way because it's not using a lot of the information. You have plenty of Petri dish that have counts that you could count, and you're just throwing them away saying these are just the guys I don't count, which is a pity. So I think it's much more elegant to measure, to actually do a proper fluctuation test analysis. But the P0 method is a very robust option, and it's using directly the exact computation we just done here and the property of the Poisson, which is it's E minus the rate of the Poisson. So you can see how switching from all the population genetics we've done to putting multiple mutants is just integrating over a space of growth rates. So there's nothing special with this. What is special is to work out what these integrals are for given model of mutation effects, of course. So we'll talk about some mutation effect models in the next lecture.
One short question, but in the Luria-Delbruck, there's no fixation probability taken into account, no?
No, because so in the Luria-Delbruck, they consider, from what I understand of it, they consider two things. First, they consider that B equals 0 in the stress. So they do not model DN. They do not model any colony that could arise while the cells are dying on the Petri dish.
Okay.
They just say whatever is a colony on my Petri dish comes from my culture that I plated on there. So they consider that the birth rate is 0, that somehow the mutation rate during the decay is not contributing. And second, they consider that every R is an ER. So they don't, because in fact, what we're estimating by the P0 method is not UR, but we're estimating UR P bar, which is the mutation rate resistance times the average establishment probability. And why? It's because to form a colony, you start from one cell and it does divisions and dies too, of course. There is death happening because there's antibiotic on the Petri dish here, there's antibiotic. And it does it enough to become a visible colony, which is like 1 million cells. So it's really, this thing is really an established population. Started from one copy and made it to become a visible colony. You have other cells that just do 2 division and die. And these are not visible. They are not colonies and they are not counted. So the colony is really an established. So to me, it's really an ER, but it's not. Usually it's considered, there's a description in the statistics of this, which is plating efficiency, but it's considered to be independent of the mutant. It's just a plating efficiency is just how many cells we retrieve when we plate a given number of cells, and somehow we lose them. So you could consider that P bar is what people call plating efficiency in the fluctuation test statistics. But the problem is that you cannot estimate it once, like you could estimate it on your permissive medium, like saying it's a problem of the way you put your cells. Some are lost, and if you estimate that, it's not going to be the same at all in the stress environment, because in the permissive medium, it's a pure birth process, so there's no one dies. P bar is 1. But in a stressful environment, and as you increase the stress, this becomes lower and lower. You have much more cells that are lost.
But Guillaume, would that not be very low for a resistant mutant that is able to withstand the antibiotic? Isn't that the logic there?
It depends on what you're considering. What is the resistance? For example, let's take a birth-death process. So for a birth-death process, you have a rate of birth B and a rate of death D. By definition, your wild type has B less than D because it's decaying, and the growth rate is B minus D. So the growth rate of wild type is this. So that's a good way to analyze it. The establishment probability for a guy that has birth rate B and death rate D in the simple birth-death process is B over D. That's the probability that from one cell you produce. Sorry. It's B over D plus B. That makes a difference. It's the probability that from one cell you produce a visible colony. So it depends. If you say that your resistance is so good that from a wild type that had a birth rate lower than its death rate, it managed to become a guy with a B that is not 0 and a D that is 0. So it has no death whatsoever. It's a purely resistant. Then this is 1. But whenever it's not true, this is not 1 at all. For example, in my experiments, we use cytox, which is a marker of dead cells. It marks some, depends on the way you die. I don't know how, I mean, we are 10 minutes over the timing, so I don't know if it's a problem.
Yeah, we should
Maybe
probably
the student,
yeah.
the students have gone anyway, but I use cytox, so it's marking the dead cells. And when I do that, I'm following with luminescence, I'm following the live cells, and I get something like this. I will maybe show those next time. And I also have the CTOX that is showing me, so that's a case where I have a rescue, of course. And the CTOX does something like this. So you have first a jump in the CTOX here, which is corresponding to the death of the wild types that are going down, right? And then if you have no rescue, it goes this way. So that's what would happen if I get a well where there's no rescue. And if I have a rescue, it goes this way. I have the parallel thing. I should put it in, I don't know, how should I put it? In red at least. That's the same phase. But then when I have this growth, I have another bump of cytox. What this tells me, of course, is that it's those guys here who are doing dead because the others are doing nothing, right? So there's a huge amount of dead cells produced. It's just that B minus D is positive. That doesn't tell me that D is 0. So whenever B over D plus, so whenever this is happening, whenever there's some death, you have a probability of establishment that is less than 1. And more importantly, it's going to depend on the stress. So it's going to be dependent on whether you're in a strong stress or in a weak stress. For example, say my line is both resistant to a high dose and a low dose, but in the high dose, it grows less fast. And it could be that its death rate is increased, right, where the birth rate doesn't change, for example.
So it's really because the establishment is a function not of competition or selection. Really is just hard selection, where you're dying just because the antibiotic is stressing you out and you're not able to divide.
Yeah, the establishment here is, but it's really the same. I mean, it's the same, exactly the same math as the establishment probability of population genetics.
Yeah.
It's just that for population genetics, the role of death is played by the fact that you are in a fixed population where you are constrained to be in a fixed number. So you die because of the others. It's what happens in the Moran model. Right. The Moran model is exactly do this. It's a birth-death process where the death rate is exactly compensating and killing you by 1 over n. Right. There's a death rate of 1 over n, if you want. In here, the death rate is produced by the actual environment without having this density dependence. Whereas in a typical experiment of bacteria that you grow and select, the death rate is here, in fact. There's no death happening in the growth, and the death rate is just because you're diluting out the guys. So they're dying out of this. Yeah. There is a death rate. It's just not the death that you would see from cytox. Yeah. But the math is really the same. You have dP equals SPQ dt plus PQ over any dBT. That's the SDE for the not here. Sorry. That's the stochastic differential equation for diffusion, where you have these fixation probabilities. And then if you go in the Feller approx of this, you're just saying that you are considering a population that starts at a very low frequency, and that has an S over NES that is large enough that whenever it avoids loss when rare, it will go to fixation certainly. And then if you do that, you have a dP equals SP plus P over any dBT approximately. That's really the Feller approx that we use. Kavita knows this. So that's how you compute a 2S. You can get a 2S probability of fixation for that, and from one copy. Stop me if I'm saying it wrong. I think it's right. But if you're doing stochastic growth, you're looking not at frequencies, but at absolute number, and the failure approx is like this. It's just really the same thing. So, and the sigma here, in the case of an approximation for a birth-death process, would be equal to the sum of B plus D. So this is the noise of demography, and this is B minus D. This is a deterministic trend. But really, you get established because you have noisy demography, and there's an absorbing state. Just the only absorbing state is 0, not 1. But it's not very different from the. I think it's not very different from the mathematical spirit of it.
Thank you.
Yeah, so it was great, Guillaume. So we'll continue tomorrow.
Yeah. So maybe a question for tomorrow. I have to make choices. So you tell me what is the best, what you want between the two. I was hoping to do the multiple rescue steps today, and I didn't. So that I could do some phenotype landscape Fisher's model things tomorrow.
Let's do phenotypic. Yeah. Right?
So you want a phenotypic. So I won't talk at all about two-step rescue.
Yeah. So I think because I think people can get some idea, at least one step they know. So therefore it's sort of more extension, perhaps a bit more involved, but maybe you can give a reference for the multi-step and start with the phenotypic one so that people are exposed to different ideas. So I think that would be nicer. But please do send us the references and if there's any additional material.
Okay.
Okay. Yeah. See you tomorrow.
See you. Bye.
Bye.